Okay, I'm back here to continue with blood. So let's get back to our slides here. Okay, so we've established that we have about 4 to 6 liters of blood volume, depending on the sex. We know about hematocrit, about 55% worth of plasma, 45% of the formed elements of this tissue that we call blood. Yeah. So I'm going to look now at the sort of the developmental process. Just like you were born as a child, you went through a developmental process to become an adult. Blood cells do the same, and they do it in different ways depending on the type of blood cell. So we talk about hematopoiesis, right? Blood, blood. Poiesis is basically the generation, the formation of it, right? So hematopoiesis, or sometimes known as hemopoiesis, the formation of blood. This will occur in the red bone marrow. So if you remember back to 2401, you looked at your bone tissues, your different long bones and flat bones, all that mess. Well, we're looking at the formation, the where, right? Where does hematopoiesis take place? It takes place in the red bone marrow. Why is it red? Well, because we're forming blood cells in there, the erythrocytes. So all blood cells are derived from a single population of stem cells called hemocytoblasts. So again, going back to 2401, what is a blast? A blast is the opposite of a clast. So a clast is something that will destroy tissue or cells. A blast, something that will create tissue or cells. So hemo, blood, cytocell, blast, to form. So the process in which we form blood cells, where it happens in the bone marrow, how it begins with this type of cell, right? This type of process that we call the hemocytoblasts. Now, I don't know what you know about stem cells at this point. Stem cells are sort of generic cells. They have the ability to become any of various types of cells in the body. So they're what we call undifferentiated at this point. They have a lot of flexibility in the idea that they can become a nerve cell, they can become a blood cell, they can become a muscle cell, a liver cell, a kidney cell. Well, these hemocytoblasts they're stem cells, but they're not necessarily the first types of stem cells. This hemocytoblast is already committed to becoming a type of blood cell. It doesn't know what kind of blood cell it's going to be yet, but we know that hemocytoblast is not going to become a kidney cell or a neuron, or it's not going to become a cardiac cell. It's going to be some form of blood cell. Depending on hormone regulation, depending on the needs of the body, of genetic instructions, different enzymatic cues, it's going to get the signals to become eventually these formed types of elements. But again, let's look at the process from hemocytoblast. It now becomes a committed cell. So at this point, it gets the signal, hey, we need red blood cells. You're going to become a red blood cell. All right, right away, so eliminate. And it starts to undergo the developmental process to become a red blood cell. So we go through different stages: the proerythroblast, early erythroblast, late erythroblast, normal blast, reticulocyte, and finally the erythrocyte. Now, you can memorize all that if you want. I'm not going to really ask those details, but I do want you to look at this part right here. This is the most significant of this developmental pathway. So if we know the significance of a nucleus in the cell, so the nucleus commands the cell, controls the cell, allows the cell to undergo mitosis, to grow, to repair. Well, what happens to the nucleus here? The nucleus is ejected. So that nucleus is kicked out of the cell. It would be like your body just kicking out the brain. No, we don't need the brain, just get rid of the brain. So it's an odd type of event that happens here, right? So we have a large cell with a large, huge nucleus. And then eventually that nucleus gets kicked out. So what happens to the cell? The cell kind of deflates, like just a deflated sphere, because it doesn't have all of that important information inside. So we have then this little deflated cell that we call the erythrocyte, or the red blood cell. So erythrocytes, RBCs, red blood cells, gives you the shape, sort of the size. We call them biconcave discs. They're concave this way, they're concave this way, right? So if you've ever gone to the meat department at Albertsons or somewhere, and little kids have been there before, they put their big thumbprint in the meat, right? In the hamburger meat. It's kind of what it reminds me of. Those little Smarties candies that are like biconcave on both sides, thick on the edge, thin in the middle. So this gives these cells a great amount of surface area. Since they have no organelles, they got rid of all the nucleus, mitochondria, all that, that is going to come at a bad consequence, right? They don't live very long. They can't repair. They can't generate energy. So once they've committed to becoming an erythrocyte, that's it, right? So you do your job. You're going to transport oxygen for as long as you can, which is not going to be too long. Say, roughly about 3 months that an erythrocyte can survive. So their cytoplasm is just occupied by this very important protein that we call hemoglobin. Right? Hemoglobin is going to be the molecule that actually transports the oxygen. So I'll address that shortly. And there's the ranges, right? So ranges per microliter of erythrocytes, right? So let's look at this protein, this hemoglobin protein. It's a red protein, thereby giving these cells that reddish color. It's involved with transporting oxygen, but it can also transport carbon dioxide. So it's a gas transporting protein. Hemoglobin is comprised of 4 chains, 4 globins, and then 4 heme components. So these are going to be the anatomy of the hemoglobin. I'll show you a picture shortly. And these are the ranges of hemoglobin within one erythrocyte. Let's go back and address the hemes and the globins. So here we have different kind of colors. We have like a tannish and a darkish brown, but these chains would represent amino acid chains, right? These are proteins. In the center of this big sort of tertiary protein, we then have this red circle that we call the heme group. Right, so there's a heme in the center of the globin, a heme in the center of the globin, a heme in the center of the globin, and a heme in the center of the globin. Now in the center of the heme group, like a bullseye, we have iron. Fe stands for iron. It's a little element of iron. Iron is very, we say it has a strong affinity for attaching to oxygen, right? So think of like a magnet. Oxygen is very electronegative, and iron is really good at attracting sort of this molecule to it to have a very, very good connection to attract and to hold oxygen. So thereby each hemoglobin, right? So 1, 2, 3, 4 repetitive parts make up one hemoglobin, and one hemoglobin has 4 hemes. Each heme has 1 iron, so each hemoglobin has 4 iron molecules within the heme to attract 4 oxygens. So each hemoglobin can carry 4 oxygen molecules, and that's an important number as we continue on. So backing up here, so hemoglobin HGB consists of 4 polypeptide globins, and in the center of those chains are heme groups. And in the center of the heme group, we have that iron atom. And again, that is the, the molecules all packed within that red blood cell. So basically a red blood cell is a little bag of hemoglobin. A red blood cell is a little pouch carrying hemoglobin, which is carrying oxygen throughout the body. So we mentioned they cannot repair themselves. They cannot generate their own energy. So their lifespan is quite short. So about 100 to about 120 days is the length of a red blood cell's life. Yep. banged up as they're going throughout the body, getting hit against blood vessels, compressed within, the body. And again, they can't repair themselves. So I don't know if you've ever felt a little bit tired. See, I haven't done much, but I feel tired. Well, on a daily basis, you're producing about 100 billion new cells per day, right? 100 billion new red blood cells per day, because they can't repair themselves. Our bone marrow has to crank these out on a regular basis to keep our normal blood volume. Now, how does this get stimulated? Well, red blood cells are produced in response to the hormone. And I will ask you this on the test. So test question, ding, 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 right? Important hormone, erythropoietin, right? Erythropoietin is the hormone that triggers the red bone marrow to kind of develop these red blood cells. So let me back up here. So erythropoietin is then going to be the trigger for this hemocytoblast to commit and to go through the whole developmental pathway to form that red blood cell. So important hormone. And it may be a little bit interesting, but you think, well, which organ would regulate erythropoietin? Maybe the heart's pumping blood, or the brain needs a good blood supply. May surprise you a little bit, but it's actually the kidneys. So your kidneys are the regulators of blood volume. So if the kidneys sense that there's not enough blood volume, that there's not enough hemoglobin cycling around the body, it triggers this hormone to get the hemocytoblast to make more erythrocytes here, right? So an interesting link there. So erythropoietin is produced in response to hypoxia. So if there's not enough oxygen carrying ability, so low oxygen carrying capacity or carrying ability by the blood, the kidneys then stimulate the release of the hormone, and more red blood cells are produced. So how does this happen? Well, there might be an accident. You might have had a big laceration, a lot of bleeding, a large hemorrhage event. So red blood cells dropped. Hey, we need more blood cells. Erythropoietin is released. Blood cells increase. Maybe you have enough cells, but maybe your hemoglobin is deficient. You may have heard of people that are anemic. Well, maybe there's not enough red blood cells. Hemorrhage, that's okay. If there's enough red blood cells, then maybe the hemoglobin is deficient. So we start to make more cells. Hopefully those new cells have the adequate amount and the correct form of hemoglobins. This can be a natural process in low oxygen level environments. If we were to go up to Rio Doso for, stay up there for a week or so, at first, it's going to be hard to breathe because it's higher elevation. We go up to Ski Apache, something like that. If you go up to Denver, you go climb these tall mountains. That basically just sitting there, your body is going to be faced with lower air pressure, and we start to have a lower oxygen carrying ability by the blood. So our body naturally will create then this hormone to create more red blood cells. If you know anything about training, if I'm going to train for a marathon, I can train in El Paso, but if I can go up to higher elevations, I'm doing the same amount of work, but my body's responding by producing more red blood cells, which can transport more oxygen. More oxygen means that I can endure aerobic activity longer, which should give greater athletic performance here, yeah? So one of the champions of this, maybe not in a legal way, but one of the champions was Lance Armstrong. So I'll show you a video of some of the illegal stuff he did, but again, using a natural process, but it's going to be sort of used in an illegal way and abused in a way that's not supposed to be used. So in the year 2020, and for a while now, we have known about this hormone, and this hormone can actually be synthetically produced. So yes, we can extract it from a human, or we can study it biochemically and then synthesize it in the lab. So theoretically, you don't have to go up to Rio Doso to train. I could just start to inject this artificial hormone, this synthetic hormone, which triggers then that erythropoietin to be released, or I should say, it triggers in the response for red blood cells to be formed. So high altitude training, you want to do it the right way, the fair way, the legal way. You just deprive your body of oxygen. You go to high elevational areas, and again, the body will respond to make more red blood cells. I'll show you a video on this and this idea of blood doping. Quite ingenious. It's not rocket science, I guess, but I don't know. It's just do it the right way, I guess. Don't become infamous for cheating. I mean, that kind of stuff. But I'll show you a video of that later in the blackboard here. So going back to our hematocrit, this is what our hematocrit should be: 45% plasma. I should say 55% plasma. Sorry, 55% plasma, 45% erythrocytes. So this is a normal blood smear. Now, this is after training in elevation or after utilizing this erythropoietin. We have increased our red blood cell count. And again, this can be done naturally. This is not illegal. This is a natural response to high elevational training. Yeah? But what do we notice? The body has responded by increasing the number of red blood cells. So we're no longer 55 and 45. Wow, we have now a huge increase in the hematocrit for red blood cells. So much greater oxygen carrying capacity of the blood. That's good. We can carry more oxygen. Side effect then is now the blood is thick. Think of it almost like a syrupy blood. So that heart is having to pump this thick, syrupy, very viscous blood throughout the body, which is, again, not very good. It's not something that we ideally want. But again, if oxygen transport is the goal, this person has achieved high amount of oxygen transport because of elevated red blood cell count. We have elevated red blood cell count. We say that this person has a condition, polycythemia. Polycythemia vera, or just polycythemia. This is an increased amount of red blood cells. And they label it here as a disorder. I don't think that's actually correct. It's not necessarily a disorder. It can be if there's a problem with the kidney producing too much erythropoietin, but normally polycythemia is it's a response to low oxygen environments, right? So it's an abnormally high number of erythrocytes. Again, can be naturally caused, can be caused by abuse of erythropoietin as well. But again, blood volume almost twice the normal amount. So we have a hematocrit, a red blood cell pack count almost double, right? So again, it puts a strain on the heart to pump this thick blood. Again, there's your numbers, if you like numbers, ranges. So on the opposite side, so we have polycythemia, too much red blood cells. On the opposite side, we have anemias. So anemias are conditions in which the blood has a lower than normal oxygen carrying capacity. So the obvious or most easy way to acquire this is low red blood cells. So the person has had an accident, they bled out too much, there's something destroying blood cells, the blood cells are not being produced properly. For whatever reason, the red blood cell count is low, which means the oxygen carrying capacity is also low. We can also have normal blood count, but we have low hemoglobin. So the hemoglobin is not in the right amount due to protein deficiencies, or the hemoglobin might be okay, but the iron in the middle of that heme is not sufficient. So maybe there's an iron deficiency. So there's different ways of becoming anemic: low blood count, low red blood cells, or normal blood cells, but low amounts of hemoglobin, or kind of not as common, it's more rare actually, the idea of abnormal hemoglobin. This typically is linked to genetics. So let's say the person has normal amount of red blood cells, normal amount of hemoglobin and iron. Well, they can have abnormally shaped hemoglobin in which the heme is kind of more hidden and is more difficult for oxygen to kind of fit its way in there and attach to the heme or to the iron there. Yeah? So thalassemias, if you can trace your blood lineage back to the Greek islands, if you have Greek descent, there is a genetic issue with abnormal hemoglobin. Some of you may have heard of also the sickle cell anemias. Used to be linked specifically to the African American population, but it's much more diverse now. But again, it's just a simple recessive condition in which the blood cells are not formed properly. The hemoglobin is not formed properly and causes then these cells to break, to sickle, and they don't maintain their integrity as blood cells. Right? So again, genetic conditions, not as common, causing anemias. So again, to summarize, polycythemia is too much blood cells, too much red blood cells. Anemias are too little red blood cells or normal blood cells, but abnormal numbers of hemoglobin or abnormal shape of hemoglobins, that kind of stuff, genetically caused. All right, so again, that will conclude our discussion on the erythrocytes. I hope that made sense. So let me convert this video now, and I'll continue working on the rest of the chapter, white blood cells, platelets, and I do want to link in some videos that I think you might find interesting as well. Yep. So you'll have a good evening, and I'll be continue lecturing and posting today and tomorrow and Wednesday lectures. Yep.